So we left it at sometimes loci are partially linked, right? And what that means is when a dihybrid individual for those partially linked loci is getting ready to make gametes, in most, if you will, of the meiosis, the homologous chromosomes are gonna pair against one another across the metaphasic plate. And when those homologous chromosomes separate from each other in meiosis 1, now a pair of alleles will go one way and another pair of alleles will go the other, right? And that's what makes these alleles, that's what makes these alleles linked, right? Because now they're traveling together. They came together from one of the original parents, landed on this dihybrid individual, but then when they need to go travel themselves into gametes towards making the next generation, they will travel together. We say they're partially linked though, because every once in a while, in prophase 1 of that meiotic 1 division, there's gonna be this thing that we call crossing over, where two chromatids from homologous chromosomes, not sister chromatids, but two chromatids from the two homologous chromosomes in a pair will exchange places, swap information, and now you have uncoupled those alleles from each other. And it used to be that the recessive A always traveled with the dominant B from one parent, and the dominant B always traveled with the recessive A. I just said the same thing twice, the other way around. But now they've reshuffled, and you have some gametes that will inherit a recombinant allelic combination. So some gametes will be recombinant, most gametes will be parental. And we finished our last lecture introducing the concept of recombination frequency, and that is the number of recombinant gametes over the total number of gametes that an individual produces. We also said that in the special case of test crosses, when you're having these dihybrid, trihybrid, tetrahybrid individuals cross or mate with nothing but recessives, homozygous recessives for all the loci involved, saying the number of recombinant gametes, it's like saying the number of phenotypic classes in the progeny, right? Because each of these recombinant gametes will generate a different phenotype when these gametes fertilize with gametes from the test cross individual that has nothing but recessive alleles, right? All right. So a few fun facts about recombination frequency. First, it's inherent to a pair of loci. What that means is if you run the same cross, but in one case you get a progeny of 120, like in the first example, but then you run the cross again, and for whatever reason you get way fewer pups, about half of them, 60. If you calculate the recombination frequencies from each of those crosses, you should get the same number. Because recombination frequency doesn't have to do with a specific mating event. Recombination frequency is inherent to how close or far away from each other the alleles are in DNA. And that doesn't change whether you have 120 pups in the progeny or 10 pups in the progeny, okay? Another fun fact, it's nearly additive. What that means is if your recombination frequency between locus A and B is 0.15, and the recombination frequency between locus B and C is 0.07, more often than not, aside from a few exceptions that have to do with weird chromosomal locations and whatnot, the recombination frequency between the two extremes, in this case A and C, would be the sum of both recombination frequencies between them, right? So in this case, 0.22. I'm going to bring you back to when we were discussing that when Mendel's laws or Mendel's work was rediscovered, there was this reborn interest in genetics and the laws of inheritance. And one of the things that was new from when Mendel was doing his experiments is that people had started seeing chromosomes under the microscope. And that there was this famous lab at Columbia University, the Morgan lab, that worked with fruit flies, and they noticed Their interest was in genetics, right? But they noticed that chromosomes behaved very much like Mendel had postulated his particles of inheritance worked. So they kind of made that connection, and they said, well, maybe these particles of inheritance that Mendel proposed travel in chromosomes. Cool. So hold that thought. Another thing they saw under the microscope, if they looked at enough cells under the microscope, is that sometimes these chromosomes, when they were preparing to separate in meiosis 1, just before they would go and align on both sides of the metaphasic plate, they would do this funny thing of crossing chromatids with one another. So at the time, it wasn't totally obvious what that could mean, but in time, the Morgan lab started putting it together. They saw that there were certain loci that were partially linked, just like Bateson, Panet, and I can't remember, and whatever, had seen with those plants. So they were seeing it with flies, and they were seeing under the microscope these funny shapes of chromosomes, apparently interchanging information. That's the best way that they could put it, right? So they came up with this idea, with this proposal, that recombination frequencies correlate with the distance between loci in a chromosome. Exactly what you just said. The further apart two loci are in a chromosome, the higher the chance that a crossover event might happen between them. And the higher the chance a crossover can happen between them means the higher the chance that they would recombine during gametogenesis, leading to one of these recombinant gametes. If these two loci are so close to each other that the chance of a crossover event falling between them is very, very, very small, you may never see a recombination event. No matter how many plants you cross, how many flies you cross. They're just so close that the chances of one of these that happen randomly across the chromosome falls right in between them is very low. And what do we say then? That those alleles or those loci are fully linked. If they start becoming further apart, and not as in them moving away from one another, if they happen to be in further apart locations of DNA, now, slowly but surely, the chance that a crossover event, a random crossover event might fall between them starts increasing. And that will increase your chances that you do see recombination events, and that means that recombination frequency will start growing. Okay? These people in the Morgan lab developed this idea and then started backing it up with a ton of experimental observation, to the point that people were like, okay, sure, you guys are right. Like, it's just like you say. So in honor of that, what used to be a 1% recombination frequency turned into what for a while people started calling a mapping unit. The higher the recombination frequency, the further apart loci were, meaning the more mapping units between them. Let's say that two loci had a recombination frequency of 5%. In jargon, people would say that they were 5 mapping units apart. If two loci in the same organism were 10% or had a 10% recombination frequency, we would say that they had a 10 mapping unit distance between them. And in honor of Morgan, that turned into what nowadays we still call centimorgans. This is not centimeters because it's not a physical distance. It's just the relative distance within the chromosome that will make those loci more or less prone to being recombined by a crossover event, right? So there is no physical correlate with this. This is just based on how often a crossover event will land between these two loci, and therefore how often we might see them recombine. And they're all 1 to 1 to 1. So 1% recombination frequency equals 1 mapping unit equals 1 centimorgan. So since those days in the Morgan lab at Columbia, this idea exploded that now you could map the location of different loci in different species by simply calculating recombination frequencies between alleles from those loci, right? So the idea was we see A and C recombine very often. Why? Because they're very, very far apart. A and B recombine sometimes, B and C recombine sometimes. All of that jives well with these maps that we have here, right? Sometimes you can derive maps from the recombination frequencies, but one thing that you need to remember is that those maps will be symmetrical. So in the first case scenario here, we see A and C recombine very often. That means they're very far apart, right? And then we see A and B sometimes, B and C sometimes. Okay? So B could be in between A and C, right? But this same map could be C, B, A. And the recombination frequencies that you would observe for each of these pairwise comparisons or pairwise calculations of recombination frequencies would be the same. C and A would still recombine very frequently, C and B only sometimes, B and A only sometimes. At the time we thought, and now we've in a way confirmed, are random, right? Where they fall is pretty random. There may be areas where they're forbidden, but for the most part, they can happen anywhere randomly. So if I were to place this iPad on the ground and then toss a coin or something and see where it lands, there is a relatively high chance that it's going to land anywhere between A and C, okay, right? But the chance that it lands between A and B or B and C is smaller because the space for that to happen is just smaller. In this case here, they're not any closer than A is to B. Okay. And that's by design, so I meant it that way. Just because these two are dominant and these two are recessive, that doesn't make them any closer. Okay. The idea here is that you need that sort of heterozygosity across the loci, and then you need to do it either in a test cross, or it would be nightmarish to do it through a self-cross. But let's say we do it with a test cross. What you're looking for, you know that in a test cross, no crossovers, this individual will have progeny that has dominant A and then recessives B and C. That's one parental phenotype. And the other half of the progeny will have recessive A phenotype, dominance B and C phenotypes. But you can count in the progeny any recombinant phenotypes that you see, like dominant A with a dominant C now. If you see an individual like that in your progeny, now you know that there was a recombination event, and then you count it as one. And you add up all of the recombination events, divide them by the total progeny, you have your recombination frequency. No, it's on the horizontal axis. So closer to one another would be this, where A and B might recombine by very rarely, and A and C very often. No, it doesn't. So they could be both dominant up here and both recessives down here, or they could be one of each. What I was trying to say is that the recombination between the A and B locus, forget dominant and recessive for now, would be very rare. If you take this individual, and once again, you cross it to a test cross individual, let's say that B codes for, again, flower color, right? And recessive is blue, and dominant is red. And A codes for stem length, and dominant is long, and then the recessive for stem length is short, right? You shouldn't think so much in terms of whether the phenotype is recessive or dominant, if that makes them closer or not. What matters is whether this combination, blue and long, and this combination, red and short, which are the parental combinations, ever change. Do you see in your progeny any flowers that are blue but with a short stem? And do you see any progeny that has red flowers with long stems? Because if you see any of those are recombinants. And the chance of you seeing any of that in this particular conformation is lower. Because if, again, this trick of tossing the coin, if I toss the coin against this backdrop, the chances of the coin landing between A and C are much higher than it landing between B and A. Will it ever land between B and A? Yeah, it will, but it'll be a lot less often. So Morgan's work triggered this explosion in what became sort of almost like an arms race to build all of these genetic maps, right? And what we mean by genetic maps is try and determine the relative position of different loci for characters that we're interested in along chromosomes. To the scientists in us, that could sound like important, interesting, and whatnot, but at the time, what was really important for folks doing these things is that if we had a better map of where loci for different characters of interest were in the chromosomes of different species of interest, then it would be easier for, say, farmers to determine if they could ever get corn with long stalks and blue grains, for instance. Could that even be possible? If they found that these loci were fully linked in such manner that it's impossible to recombine blue grains with long stalks, then they would give up. Does that make sense? Yeah? So there was certain interest in determining how information was mapped in a way in these chromosomes, because that correlated directly with what was possible. All right, cool. So how do we do this? Let's say that they give us these recombination frequencies between loci P and Q. They say that it's 0.10. That's expressed as recombination frequency. So that would be 10% or 10 mapping units. The frequency between Q and R is 0.15, 15% or 15 mapping units, and 0.05 for P and R. So what you do is you build a puzzle. That's what you do. You build a puzzle. So we start one by one. Could it be like this? Could the map be like this? So then what you do is you say, okay, P and Q, 10. All right, I'm going to write it down, 10. Q and R, 15. Okay. So since recombination frequencies are nearly additive, the distance between P and R should be 25 mapping units, right? Is it? No. So we can rule out this particular map. Could it be this map? Well, let's see. P and R 5, so that checks, they're close. Q, R and Q, 15. So the distance between P and Q, bless you, should be 20. Is it? Nope. It's this. So literally, what you do is you start building these maps, and that's why it's so important for you to have scratch paper and use it during the exam, because you'll start building these maps and see if it makes sense with the data that you have, right? There may be some questions in your exam that look like this. I will give you a list of recombination frequencies, and you have to come up with a map. There will be some questions that I give you the recombination frequencies, and the options are different maps, and you choose the one, right? So always variations on the same thing, that you build these puzzles and see which one makes sense with all of the available data. Now, I say that the map could be this, or what else could the map be? I see some people going like this, right? Yeah, exactly. That's exactly right. It could be Q, P, R. Now, the relative distances should be maintained, but we can't rule out the possibility that they're oriented this way. In real life, it wouldn't be one way or the other. In real life, it would be one way, but we need some sort of point of reference, and that point of reference could be the end of a chromosome, it could be a centrosome, it could be some sort of location that is invariable in a chromosome. We don't have that here, so right now these two maps are symmetrical and they're both valid. Now you try. Okay, I'm assuming that maybe you go through at least one of them. Yeah? If you didn't, you can finish them later, but one thing that I wanted to do is share with you a trick. I'd make this a little easier. So the first thing you do is you rank the recombination frequencies in descending order. You list the highest one first. Why is that? Because what that tells you is what are the loci on both ends of this map. right? So the furthest apart. So in this particular case, A and C are 22 map units apart. Now we know if A and C are the ends of this map, we know that B will be somewhere in the middle. So what you do is then you quote unquote move the middle locus until you match the distances to the outer loci. In this case, we would go something like this: A to C, 22, and then let's see if this works. I'm gonna throw the B in there. Let's see if it works. And then I move it. Is it moving? Wow. Right? So I know that B is really close to C, so I will place it somewhere there. And then I say 7 mapping units here, 15 mapping units there. What about tri-hybrid test cross, 3 loci? How do we map 3 loci at a time? We just did it, right? If you want to map 3 loci like A, B, and C, MNO, P, Q, R, right? You do 3 independent 2 loci crosses or test crosses. Then you figure out the relative recombination frequencies between any 2 pairs of loci at a time. And then you do the little maps like you just did. In practice, what happens is that means setting up 3 times the number of crosses. And it came a time during the history of science that it was easier to set up 1 cross and do it all at once. And that's where the tri-hybrid test crosses came about. So the idea here is we have now a triple homozygote recessive mouse shown here. We have seen this fellow before. White coat, long tail. Sorry, this guy here. Short tail, white coat, and curly whiskers. We can barely see it, but apparently those whiskers are curly. You gotta believe me. Right? That's our triple homozygote recessive test mouse. And then we cross it to a mouse that is tri-hybrid. It's heterozygote for the 3 loci that we're interested in, that we want to map relative to one another. So there's going to be a lot of progeny. Obviously, you don't do these once. No pair of mice will give you so many. Well, eight maybe, but if you really want to crank up the numbers, you need several of these crosses, and you amount progeny in the hundreds, right? And once you do, these are the steps that you take. It's very, it's a protocol that you follow, okay? It's a series of steps that you need to remember and practice. First, you need to find the parental gametes. You know that these loci are partially linked, otherwise you wouldn't be doing this. So you know they're partially linked, and you're trying to figure out recombination frequencies at the end of the day, right? So the best way to find recombination frequencies is to find those recombinant gametes or recombinant progeny so that you can divide it by the total. How do we know what's recombinant? How do we know what's parental? That's the first step you take. You look at the most abundant progeny, and that is what came from parental gametes. Why? Whenever loci are partially linked, most of the gametes are parental, only some will be recombinant. So we go for the most common gametes or progeny first. And when we do that, we notice that the most common progeny in this particular cross has long tail, white coat, long whiskers. Okay? It would be this guy here. And then the second most common phenotypic class is short tail, brown coat, short whiskers. So given those most abundant phenotypes, you can from there deduce what the most common gametes that were being produced, and those are the parental gametes. Recessive A, dominant B, recessive C, and then dominant A, recessive B, dominant C. What you don't know yet is in what order these things are in the chromosome. You know that those are the allelic combinations, but you don't know yet what order they're in and what the distances between them are. So that's what we'll do next. Second, you have to find, you go the other way, the other extreme. You go find the rarest of gametes. what is the rarest of phenotypes in this cross. And by looking at the numbers, 5, it's kind of close, but there is a 1 here and a 1 here. So the absolute rarest of the gametes are these guys that I wrote here. Does that make sense? So you match the phenotype in the first column to the locus A, the phenotype in the middle column to locus B, and the phenotype in the last column to locus C. So long tail was dominant A, brown coat was dominant B, short whiskers is recessive C. Yeah? And then you do the same for that. So now you wrote your parentals, you wrote your rarest. What we need to find out now is which of these loci is different between them. So how do we do that? We basically look at which combination of loci are the same in parental gametes and the rarest of gametes. And in this particular case, it was B and C, right? So in the parentals, you had dominant B with recessive C, and in the rarest, you have dominant B with recessive C. The other parental was recessive B with dominant C, and the other rarest was recessive B with recessive C. So they're the same. And then there's a third locus, in this case A, that will be different. And that is your locus in the middle. Okay? So we were writing ABC because we didn't know any better. Now we have some information. Now we know that locus A is in between B and C. So we can rearrange the parental gametes with the correct locus order. So now instead of saying ABC, I'm going to say BAC. BAC. Or the corresponding flipped versions. For now, just mechanics. We find the most common. Those are the parental gametes. We find the rarest. Those are recombinant, right? But what we do is we align them, and then we look for the two loci that are the same between the parentals and the rarest, and the one that is different between them, that's going to be the locus in the middle. Why is the rarest gamete the one that has the recombinant locus in the middle? This is why. Let's say that we have that tri-hybrid individual, and like it happens often when loci are partially linked, no crossover events happen to land between any of them. There were no crossover events in this area, so the tri-hybrid individual will maintain the same allelic configuration that it originally had, all the way from the parents, right? There's no change. These gametes are the same as the ones that were being produced by the parents. That's why we call them parental gametes. Now let's say that there is, by chance, a single crossover event that happens to fall somewhere in this region. Sometimes it will fall between the first and the second locus. Other times it will fall between the second and the third. And this will happen sometimes that one crossover event falls somewhere in the region where the three are. What's going to be super rare is that two crossover events fall within the same region. And when that happens, again, all of this is probabilities, okay? Probabilities, the probability is, or what's most likely is that one will fall between the first and the second, and the other, the second crossover event, will fall between the second and third loci. So this is going to be extremely rare. And what happened here was that the only allele that changed with respect to the parental gametes is the one in the middle. Notice how for the Y locus here, now the allele combination relative to Y changed relative to the parentals. In the parentals, it was all recessives or all dominants. But now you have these gametes that have X and Z recessive, but Y dominant, or vice versa, X and Z dominant and Y recessive. That will happen very rarely, if at all. And that's why in these 3-point mapping crosses, we find those rarest of individuals. We look for the one character that is different from the parents, and we establish immediately that that's the locus in the middle. Anyways, so this explains why the rarest gamete corresponds to the locus in the middle. And now that you have the correct map, we can go around calculating recombination frequencies and therefore mapping distances between these things. And here's where it gets, I'll be honest with you, it gets a little tricky. So we know that in the parental, we had, for instance, dominant dominant B, recessive A and C, or dominant recessive B, dominance A and C, right? If you're trying to calculate the distance between B and A, the first two, what you really need to do is calculate the recombination frequency between these two. So what you do is you first establish what the recombinant gametes versus these parentals would look like that involve B and A. So if one configuration was dominant B and recessive A, you look for all possible combinations that have now dominant B and dominant A. If the other parental configuration was recessive B and dominant A, your recombinant gametes are those that will have both recessives. The second, yes, yeah, that's a nice way to put it. You fix the first locus, in this case it would be B, and then you're looking for a change in the second. Yeah, I don't think, I can go check, but I don't think there was any problem in the bank that had the table. If you have the table, what you do is you look for these things in the table. So let's look for that first one here. Where do I see dominant B with dominant A? And I see it here, and I see it here, right? So I'm going to write 17. Where do I see both recessives? Here and here. I'm circling what I already circled, but you get the point. And how many of those? Those are 12 plus 1, 13. So now, if I want to calculate the recombination frequency between B and A, I need to add up all of the recombinants. So 1 plus 16 plus 12 plus 1, that gives me 30, out of a total of 120. Yeah? So 30 divided by 120, 0.25. That is the genetic mapping distance between B and A. We do the same for between A and C. So going with your idea, we fix the first, recessive A. We know that in the parental, recessive A goes with recessive B. Now what we'll do is we'll fix recessive A and we change the recessive C to a dominant. For the other parental, we know that it's both dominant, so we fix dominant A and we change it to recessive C. And then we go around fishing for these categories in the table. We add up all of those that show up, divided by the total, and we get 0.10. So now we know that between between A and C, there is 10 mapping units. And the question in the exam might be, what's the distance between B and C, for instance? And then you say, oh, 35. Let's circle back, and yeah, we have some time. So let's circle back to this question that is, I find, kind of counterintuitive. And in my experience, it kind of can make many students reasonably trip a little. We've been saying that the further apart two loci are in a chromosome, the higher the chance of a crossover event between them, the higher the recombination frequency between them, right? So let that thought settle for a second. The further apart they are, the higher the recombination frequency. Then you'll find everywhere, all textbooks, all websites, all videos talking about these things, that the maximum recombination frequency is 50%. So why is that? If two loci are sufficiently apart, shouldn't 100% of the gametes be recombinant? Why is it that we know that the maximum recombination frequency is 50? The full linkage, so you're thinking 50-50, but 50-50 was in reality all parentals, right? When there is full linkage. When there is full linkage, that 50-50 that you're thinking of is there is two gametic classes. One with one combination of alleles, the other half with another combination of alleles, instead of having all four possibilities. that we had when there is no linkage whatsoever. Does that make sense? Yeah? A little bit? Because you said full linkage, so when there is full linkage, these things are not going to separate. My question or my point is this, what can be a little confusing is this. When they're together, like right next to each other, the recombination frequency is 0. We start separating them, and Morgan told us the further apart they are, the higher the recombination frequency will be. Why does it cap at 50? Why doesn't it keep going 60, 70, 80, all the way to 100? Why can't it be that if two loci are far apart enough in a chromosome, all of the gametes that an organism makes are recombinant with 0 parentals? Why is that? What I'm saying is, I see where you come from. So what I'm saying is, let's say that we have these parents where the dominant A lives with recessive B and recessive A lives with dominant B. If there is full linkage, there is only 2 classes of gametes that are going to be made. One parental and the other parental. That's 0% recombination frequency. If they're not linked at all, let's say these things lived in different chromosomes, there's no way that there is any linkage between them, then these organisms could make 4 different gametic types. Dominant A with recessive B, like one of the parents. Recessive A with dominant B, like the other parent. And then you would have the 2 recombinants. Dominant A with dominant B, and recessive A with recessive B. We call that 50% recombination frequency because half the gametes are recombinant, new, and the other half are old, parental. What you're saying is if they all swapped, then they're going to end up traveling to different poles anyway, so isn't that 50-50? What I'm saying is a little different. What I'm saying is, why can't there be 100% recombination frequency, meaning that this organism makes nothing but double dominants and double recessives? Why is it that this can't happen? With the crossovers, yeah, elaborate, what are you thinking? For what? For chromatids, yes. So much of it relates to that. The fact that we discussed a number of times, let me see, I can add it. The fact that you have when there is crossover events, generally what happens is there is only one of the chromatids involved. So you can have these 2 loci go further and further and further and further apart. There's 2 chromatids that are not involved in those crossover events. So that's part of the explanation. No matter how many crossover events you can have between these 2 loci, there is 2 chromatids that are never touched. So that's part of the explanation. The other one, going back to what you had in your problem on Tuesday, there can be instances in which all 4 chromatids are involved in a crossover event, but those are rare. So for any time that this happen, meaning that you're going to have 2 crossover events between 2 different pairs of chromatids, for every time that very rare thing happens, there will be an equally rare thing happen, and that is there is no crossover at all. These things tend to, in large numbers, balance one another. So this phenomenon here is very rare, so is this. But just like one rare thing can happen once in a while, another rare thing can happen once in a while, and they cancel each other out. And the other explanation is, one might say, well, hold on a second. If I separate 2 loci enough, this phenomenon can happen more often. It's rare when they're close, but as they're going further and further and further apart, it could happen more often, right? So what happens in practice, and this has been confirmed experimentally, is that you may have a crossover event between these 2 here, and then a little later on, you have a crossover event between those 2 there. If we stop there, that would make for 100% recombinant gametes. Now, 100% of your gametes have an allelic combination that is different from the original. You will have 100% of the gametes with 2 dominants or 2 recessives. But the further apart these things are, the higher chance that there's going to be a third crossover event here that undid the first. And maybe even a fourth crossover event here that undid the second. So in practice, what happens is as loci get further and further apart on the same chromosome, crossover events between them start accumulating, and they can go any direction. And when you average across all of them, you end up with 50-50. It's almost like, since this is a collection of random things, you end up with a random distribution. And what is a random distribution? Independent assortment, where half of them are going to be like the parentals, half of them are going to be recombinant. I say this because there can be an intellectual, very normal intellectual clash between thinking the further apart loci are, the higher the recombination frequency, but then these talking heads come and say, no, 50% is the maximum. Why? Because, and what this is basically saying is that as loci become, if this is the distance in the chromosome, and this is recombination frequency, when they're super close, there's going to be zero recombination frequency. When they're close but not attached to each other, very low, and there's going to be a slight lag, and then it starts going up, and then at some point levels off at 50. Because after 50, you start getting this averaging across different meiotic events that will cancel out recombinations that are undone by the next crossover. So you end up flipping back and forth, parental recombinant. You do that enough times, and you end up, on average, with 50% recombinant, 50% parental. Okay, almost there. There is one use that we make of linkage today, and that has to do with, you may know a disease, a condition that you're interested in, a predisposition to a disease, right, in the human population, but you have no idea what gene may be behind it. Right? And there will be a day, and that day is approaching fast, that we simply sequence everyone's genome, and then we compare everyone's genome, and then we group the people who have a condition, those who don't, compare the sequences, and statistically can try and figure out where those things might be. Until that day comes, because we're still far away from that statistical power, until that day comes, linkage is used in this way. Remember when I told you that there is molecular markers that are diverse enough in the human species, in human populations, that chances that all of us here have different combinations of these markers because of our different evolutionary histories? Remember when I said that? Okay. It's not easy to look at all of the genes, but we can look at the handful of markers that are different among all of us, and we can establish relatively quickly which combinations we have. So you grab any group of people, and some will be V1, V3, some will be homozygous V2, some will be V1, V4, and so forth. We did this with the paternity test and the crime scene, right? So everybody has some sort of molecular fingerprint that we can kind of quickly establish. What happens is, when you're interested in a given condition, it could be that you don't know where the gene for that condition is. But you can take all of these people with that condition and go specifically look at those markers. And it turns out, I'm oversimplifying, if you look at enough of the markers, you may land on one for which this group of affected individuals here are V1, V3, V4, V1, V2, V1, V1, V5, V1, V6. And these here are V2, V6, V3, V4, V5, homozygous, and so forth. What's common among all of these guys with the condition? The V1. Is V1 the gene behind this condition? No. Because V1 is actually some molecular marker that has to do with these short sequence repeats. It's just a molecular handmark in DNA. It doesn't code for anything. It's not a gene. It's just a molecular print, right? But what do we know about that molecular print? That it seems to be linked to whatever gene is causing this. Because every time that we see someone with the condition, they have this V1 thing. So now we homed in on what may be a relatively confined area of the genome where we can go in and look in more detail. That's why we care about linkage today. It's not just to make your life's hell in a genetics class. We do use this in biomedicine today. So knowing that a condition is fully linked to a marker will give you boundaries of where to look. Yes. All right, guys, I want to be respectful of your time. So, and this was just saying that if you, V5, no, not V5, V1. If V1 happens to be linked to the condition, now you can home in on V1 and look in the vicinity, the near vicinity, to see what gene might be responsible for.